I'd like to welcome you to Wyatt Schoeneberger's honors presentation. Wyatt's been a student of mine, well, since for a long time, and got interested in this project in applied science, and encouraged him to do not only library research on that, but also do a research experiment. And he's been very involved in this research project for a year and a half at least. They're collecting data and doing a lot of things independently. Wyatt's from Neely, Nebraska. He's graduating this spring, planning to do a master's in anatomy or physiology at UNMC and then apply to medical school. So that's his plan. With that, I'll let Wyatt introduce his topic.
I'll be talking about the influence of nutrient availability and how it alters algae growth and then the overall accumulation, specifically within green algae. Some background into why this is relevant. Every year, our global energy demand is increasing. As it increases, we're also looking for alternatives to fossil fuels that will help to reduce our carbon footprint. So these new alternatives should be sustainable, renewable, and especially cleaner. Biofuels present a strong alternative. Currently, there are 4 generations of biofuels. The first generation are traditional crops like corn, soybeans, and vegetable oils. The second generation uses biological waste, non-food plants like shrubs or trees. And then the third generation uses algae biomass or cyanobacteria. And the fourth generation builds upon the third generation by engineering the algae or the cyanobacteria to make them more efficient in producing lipids. The graphic just shows a little bit more about them. So some of these generations have limitations. Like the first generation, the farmers have to decide whether to grow the crop for food or for fuel, typically going to grow what is more economical, and that usually leans towards the food aspect. Additionally, with the first and second generations, these 2 generations require a lot of land to grow the crops. So this is where the third generation tries to improve upon these by using biomass sources that will take up less land and also not take up cropland. And some of these other generations, like the first generation, are not as optimized to produce lipids as other generations. In corn, it can reach up to around 10% lipids per dry weight. Soybeans can get around 20%, but within algae, like I'll talk about later, they can get up to 80% in some species, so that makes them a much more effective crop for biofuel production. What is algae specifically? Well, algae are unicellular or multicellular photosynthetic organisms, as we'll see here in a bit. I specifically should look at microalgae. Algae converts sunlight, water, and carbon dioxide into biomass that will be used for them to grow. There are many types of microalgae. Some main ones are cyanobacteria, as I previously mentioned, green algae, which I will be focusing on, diatoms, and then golden algae. And like I said, some of these species can reach up to 80% body weight in just lipids, so this makes them a strong crop to use for producing biofuels. Why I chose algae specifically? Well, algae has a rapid growth rate. Some species can double their body size within 24 hours. They also have the ability to grow in altered and diverse environments compared to traditional crops. Algae are aquatic, so they can grow in pretty much any type of water source. The picture shows a typical raceway farm in which the algae will be grown. Additionally, with algae, there is no food or fuel conflict. Algae would typically just be grown for fuel only. And also with algae, like I previously mentioned, they can reach up to 80% liquid weight. So that makes them a higher producing crop that would be more effective for that biofuel. So what is the role of lipids within algae? Well, lipids provide energy storage, mainly as triacylglycerols within the algae. Lipids are also a main structure component to the membrane. As triacylglycerols, they are stored as typically 16 to 18 carbon fatty acid chains. The image on the right shows a triacylglycerol structure, so it has a glycerol backbone and then 3 fatty acid chains that would be 16 to 18 carbons long. Additionally, certain lipids can be increased under certain conditions by redirecting the metabolism, which will decrease protein synthesis and then also decrease phospholipid synthesis to produce more triglycerols instead. This picture shows a simplified metabolic pathway in the algae. So we can see in the chloroplast using light, CO2, and O2 from water, we will eventually get a triacylglycerol within the chloroplast. Additionally, if the free fatty acids aren't used in the chloroplast and the cytosol, a triacylglycerol can also be produced. So once again, my research objective was to determine how the effects of nitrogen and or phosphorus will specifically alter algae growth and then also their lipid accumulation. In my experiment, I looked at two species. I looked at Colloquium and Schoeneberger species. The picture on the left is a picture of chlorella under the microscope, and then the picture on the right is our multi-cell sexy Schoeneberger, as some might say.
That's right. You got here just in time.
Yeah, I had two experiments. I conducted a 10% concentration experiment, which was a 90% reduction in the sources of nitrogen and or phosphorus. And then following that experiment, I wanted to see if an increased reduction would lead to higher lipid yields. So for that experiment, I did a 95% reduction, and I'll refer to that as a 5% concentration. The algae was grown in HC Bold's modification of Bristol media in triplicates. There was a total of 30 mils of media per 50 mil centrifuge tube. These were all grown on a shaker under 12-hour light cycles. And each experiment had 4 groups with the 3 triplets. There was a control, the reduction of nitrogen with 100% phosphorus, 100% nitrogen, 10% phosphorus, and a reduction of both. In the 5% group, we had a similar setup with the control, the reduction of phosphorus, and normal reduction of nitrogen, normal phosphorus, reduction of nitrogen, and then reduction of both. So for measurements, I kept track of overall biomass growth over the experiment using a spectrophotometer. Every week I calculated the absorbance at the 454 nanometer wavelength and the 680. These wavelengths are just associated to the chlorophyll within the algae, as seen in that graph over there. Additionally, once the growth was completed and I wanted to perform lipid analysis, I did this by centrifuging and drying the algae so I could get the algae dry weight. With the dry weight, I could then use a solvent extraction method to extract the lipids, and from that I could dry them to get the dry lipid weight. From all of these numbers, I could calculate the percent lipid per that dry body weight of the algae. So I took the dry lipid weight divided by the dry algae weight, multiplied by 100 to get the percent. This graph shows the chloral growth using that spectrophotometer to calculate the absorbance in the 10% experiment. The y-axis just shows the absorbance at that wavelength. The left is the 454 and the right is the 680. And then the x-axis is the time in weeks that took one absorbance each week following a week of growth for a total of 3 weeks. Additionally, the dots are the averages of each triplicate, and the error bars are the standard deviation. Within the 10% experiment here, we can see that the control had the highest overall growth, followed by the green line, which would be the reduction of just phosphorus, and then the reduction of nitrogen in the orange, and then the reduction of both nutrients in the light blue group. In the higher growing groups, you can see that there was a slightly larger standard deviation than the lower growing groups, but over the 3 weeks, you can see that there was growth. Additionally, in the 680 wavelength, we can see that the overall growth or the absorbance was slightly lower just at that wavelength compared to the 454 wavelength. In the 5% experiment, we see similar results to having growth over the 3 weeks. In this experiment, the reduction of nitrogen, which is that orange group, showed the highest overall growth with a relatively small standard deviation. The next highest group was the reduction of just phosphorus, which you can see had relatively large standard deviation, so some groups were higher growing and closer to that absorbance of the nitrogen group. The lowest growing groups in this experiment were the reduction of both nutrients in the light blue and then the control in the dark blue. These two groups were pretty close together with smaller standard deviations than the previous groups. At the 680 nanometer wavelength, we can see that overall absorbance was lower again, but the same trends were seen. In the Senate-Desmus, at 454 and at 680, we see growth over time again. In the Senate-Desmus, interestingly, we see that the reduction of both nutrients in this example had the highest overall growth, but once again, you can see with the error bars that the standard deviation was quite large in this group. The next highest growing groups were the reduction of nitrogen in the orange, the control, and then slightly lower, around half of the overall absorbance of the highest growing group. You can see that the phosphorus was the lowest one, and at 680 it was slightly lower once again. In the 5% group, we see trends similar to the other groups where the reduction of phosphorus was the highest growing group with a much smaller standard deviation. In the final sample, we can see that the standard deviation was quite small. The next highest growing group by a significant margin, you can see was the reduction of this nitrogen, which had a relatively smaller standard deviation once we got to the end of the experiment. The control was the next highest group, and then the lowest overall group was the reduction of both nutrients that had a much lower overall absorbance control compared to the reduction of nitrogen. And at 680, we can once again see that it was overall lower, but the same trends were seen with growth increasing over time. So once again, once the growth period was completed, I could conduct lipid analysis. Lipid analysis was done by combining the triplicates due to a limited volume. The left graph shows the lipid yields within each group in the 10% experiment, and the right shows it within the 5% experiment. The y-axis is just about lipid percent per net dry weight, and then we have our groups. So the control for the 10% experiment with chloral had around 2 to 3% lipid weight. The reduction of nitrogen had the highest of this experiment, around 15%, and then the reduction of phosphorus was a little under 5%, and the reduction of both nutrients was a little bit over 10%. Within this experiment, we can see that comparing the highest group to the smallest, the nitrogen being the highest and the control being the smallest, that there was roughly a 6 to 7 times increase in lipid yields for this experiment by altering the nutrients. In the 5% experiment, the one group that had measurable lipids was the reduction of both nutrients, or the reduction of just nitrogen. This group reached near 40% lipids per body weight. The other three groups had lipids that were not measurable with our balance. The Senate-Desmus experiment, our graph is set up the same way, with the 10% experiment on the left and the 5% experiment on the right. This time, our scale is slightly smaller. We're only going to 30% instead of 50%. In this 10% experiment, we can see that the control was around 2 to 3% again. The reduction of nitrogen reached around 17%. The reduction of just phosphorus was a little bit over 5%, and the reduction of both nutrients in this example had the highest lipid yield, just under 25% in this experiment, comparing the highest group being the reduction of both nutrients to control. There was roughly a 12 to 13 times increase in that overall lipid yield per that dry weight. In the 5% experiment, the reduction of both nutrients and the reduction of phosphorus had measurable lipids. The reduction of nitrogen was right under 25%, similar to the reduction of both nutrients in the 10%. And then the phosphorus group was under 5% as well. Comparing these two, you can see that there's a large difference in lipids between the two treatment groups, but there was no control to compare to due to not having measurable lipids. So once again, in our mine results, we can see that lipid accumulation did vary under the altered nutrient conditions. Additionally, the reduction of nitrogen typically showed a consistent effect in increasing lipid yields while maintaining a relatively stable biomass growth. Additionally, some growth was diminished. The overall growth rate was diminished in certain groups. This was typically the reduction of both nutrients, but that was not seen in all experiments, like the Senendesmus, when it had the reduction of both nutrients was its highest grown group. But overall, the lipid yields were varied. In a comparative study that looked at Colloquium that also altered nitrogen and or phosphorus, they either completely deprived the algae of the nutrient or limited it to just 1/8 of the daily recommended concentration. They found in all groups, comparing to the control, which is the far right 3 bars, that in reducing or depriving these nutrients, they had an increase in overall lipids. Additionally, in their experiment, they found that the reduction of just nitrogen and then the complete deprivation of phosphorus showed the highest overall lipid yields. Also, with their experiment, we can see that they found that when you grow the algae in a normal stock concentration of all nutrients and then expose them to the reduction, that's when you get the highest lipid yields. And that sample is expressed by the light green bar in their experiment. Additionally, they had their standard error bars, and we could see that the standard deviation was quite small within all groups. So overall, for the future direction with algae biofuels, there needs to be some improvements. Currently, with the cultivation and harvesting techniques, they're quite expensive to do, and they are not completely efficient in picking up all the algae biomass, so there's waste and profit loss. Additionally, the lipid extraction methods are not as efficient as they would like, and they are also quite expensive. So improving these methods will help to make algae a much more prospective crop for farmers to grow. And additionally, like I mentioned with the 4th generation, with engineering, whether that be just metabolic like I did, or actually genetically engineering the algae, there needs to be further research into that to determine the true extent at which they can alter the algae to make them the most efficient in producing triacylglycerols for biofuel production. I'd like to thank Dr. Hammer for being my advisor and helping me through all of my research, and everybody for being here. Yeah, thank you.
Any questions for Wyatt?
I got lots.